Good morning, everyone. Today we will be discussing the advancements of biotechnology and its applications in the world today. We will cover topics such as the Green Revolution, genetic engineering, and genetically modified organisms, GMOs. We will look at the impact of these advancements on our day-to-day lives, the environment, and society at large. Please sit back and relax as we explore this very interesting and important topic. Biotechnology is a highly dynamic area of biology that has seen rapid growth in recent decades. It involves the large-scale production of biopharmaceuticals and other biological materials, using modified microbes, cell cultures, and other living organisms. Biotechnology has already been put to use in many industries, such as medicine, agriculture, and environmental remediation, giving us the tools to create new products, address existing issues, and even potentially develop treatments for serious illnesses. Biotechnology has been a game-changer in agriculture since the 1960s, which marked the beginning of the Green Revolution initiated by Norman Borlaug. This has been immensely influential in improving food production. With biotechnology came the ability to increase crop yields, increase plant robustness, and generate superior quality products. Additionally, this breakthrough science has enabled stunning achievements such as genetic engineering and control over gene expressions in microorganisms. Such incredible breakthroughs have propelled us closer to fully understanding life and how to use it in the most efficient way. Biotechnology is a continually expanding area with extensive uses. The Green Revolution in our country is attributed to the efforts of four eminent personalities: William God, Norman Borlaug, Corona, and M.S. Swaminathan. All these individuals have made their own, respective contributions to the development of biotechnology, resulting in the triumph of our agricultural sector. Biotechnology has profoundly changed our approach to food production and the application of chemical fertilizers. Through genetic engineering, farmers can gain access to improved techniques for increasing yield and minimizing their need for expensive and dangerous chemical additions. Chromosome revolution has caused a notable expansion in the number of plants with sought-after characteristics. Modern green revolution has enabled a more productive use of resources, and gene revolution has enabled the incorporation of genetically modified organisms, creating crops with improved nutrition and disease resistance. Additionally, with the aid of modern green revolution, biotechnology can be utilized to reduce the impact of climate change on agriculture. Biotechnology involves manipulating genetic material to create useful products. Genetic engineering allows scientists to alter the genes of organisms such as microorganisms, plants, and animals in order to achieve the desired qualities and features. These modified organisms are then referred to as genetically modified, GM, organisms. GM organisms have the potential to produce products which can benefit people, such as medicines, disease resistance, and environmental adaptability, hence making biotechnology an increasingly important research area. Biotechnology has revolutionized numerous industries, especially in regards to agriculture. Contrary to popular belief, most GMO plants are resistant to pest diseases due to being specially engineered to have a higher tolerance to environmental stresses. Moreover, the use of GMO plants helps reduce post-harvest losses and elevate the nutritional value of food, making them a beneficial tool for sustainable agriculture. Biotechnology has become a vital asset in various areas of science and business. Particularly noteworthy is Bt toxin, generated by the bacterium Bacillus thuringiensis, employed for a number of years to shield crops from numerous types of bugs. It's a functional, secure, and eco-friendly approach to insect control. Bt toxin has also been implemented in the manufacture of pharmaceuticals and the progress of genetic engineering methods. These models of biotechnology illustrate the potential of this technology and its extensive range of applications. biotechnology can be utilized as an effective approach for pest control. Various biological agents, such as Bacillus thuringiensis, Agrobacterium, Clostridium botulinum and Clostridium tetani, can generate toxins that are able to destroy lepidopterans, coleopterans and other insects. Furthermore, the toxins are highly specific to target pests, making them a much better solution than chemical pesticides, as they have minimal effects on non-target species. Biotechnology is an evolving science, allowing us to pinpoint and modify genetic coding in living organisms. Bt toxin protein is an example of the progress made in this area. The protein stays dormant in organisms like Bacillus thuringiensis, beetles, dipterans, and lepidopterans, proving useful in protecting crops from infestations without affecting beneficial insects. Biotechnology and its applications have been the focus of this slide, with a more specific focus on the activation of Bt toxin protein in the guts of insects. It has been determined that this toxin protein is activated by either an alkaline pH, an acidic pH, the absence of epithelial cells, or the presence of more proteases. Biotechnology has had an immensely influential effect on medicine. For instance, the toxins coded for by the CRY-IAC and CRY2A flat genes are used to combat various illnesses. These illnesses include the corn borer, papaya ring spot virus, tomato bruise and cotton boll worms. Through the utilization of biotechnology to inspect these illnesses, specialists have had the option to create solutions for controlling, if not eliminating, them. Biotechnology has been shown to be an effective solution in managing agricultural pests without the utilization of traditional pesticides. A prime example is the gene CRY-IA flat, which is capable of producing a toxin to control multiple insects, including the corn borer, cotton boll worms, tomato bruise, and rice wilting. This reduces the reliance on insecticides and enables farmers to utilize safer alternatives. Biotechnology is a rapidly evolving area with its applications becoming more intricate in recent times. This slide discusses how Bt toxin is being employed to target certain structures in bugs. When activated, the toxin binds to the midgut of epithelial cells, as well as the labrum, hypopharynx, and mandible of specified insects. This technique has been critical in restraining pest numbers, and future research may bring forth further possibilities in this sector. Biotechnology is a rapidly evolving field with many applications in agriculture, one of which is to combat the infestation of plant roots by nematodes. A control agent can be introduced, such as Agrobacterium, Longodorus, Meloidigine incognitia or Xiphenema, which have been proven to safely suppress the population of nematodes and be safe for the surrounding environment. Through biotechnology, we can now more effectively control the spread of these pests and protect our important crops. Biotechnology is a scientific field that is evolving quickly, modifying how we investigate and relate to our environment. A good example of this is RNA interference. Found in all eukaryotic creatures, it serves as a type of cellular safeguard and works by halting certain RNA molecules, which can in turn regulate gene expression and impact the organism's development. This is only one of the several uses of biotechnology being investigated nowadays. Bioengineering and its applications have revolutionized the way scientists approach a wide array of medical and industrial challenges. One particular application of bioengineering is RNA interference, which is sourced from twofold: infection by fungi or viruses containing RNA, and transposons. These sources can be used to regulate gene expression, providing novel approaches to tackling complex biological problems. Biotechnology has enabled the manipulation of living organisms, such as nematodes, through the use of vectors like Agrobacterium, Mycobacterium, Bacillus thuringiensis, and Rhodopseudomonas. These vectors can be utilized to introduce specific nematode genes to the host, permitting the control of nematode genetics. This could be useful for the eradication of pest infestations and more productive and economical crop production. We will be discussing biotechnology and its applications, focusing on double-stranded RNA, which is used to silence mRNA of the nematode. This double-stranded RNA is composed of both the sense strand of RNA and the antisense strand of RNA, not the sense strand of DNA. Did you follow that? It is imperative to recognize the significance of biotechnology in modern society. Biotechnology has significantly impacted medical diagnostics, treatments, and research. It allows for the synthesis of hormones and other biomolecules within a lab environment. Cloning of DNA, RNA, and mRNA provides the ability to synthesize any hormone, which has potential usages in therapeutics and vaccines. For this question, the answer is options 2 and 3: cloned RNAs and cloned mRNAs. Biotechnology, combining biology and technology, has seen increasing use for the creation of new medicines and treatments. Already, 30 recombinant therapeutics have been approved for human use worldwide, taking a more tailored approach to treating various conditions. These therapeutics draw on the latest developments in biotechnology, such as gene therapy and cell-based therapy, to supply novel and improved treatments. Biotechnology has had a significant impact on the medical field in recent years, primarily through the production of recombinant therapeutics. These treatments are created using recombinant DNA technology, and in India alone, there are currently 15 such therapeutics marketed, providing effective treatments for people living there. Ongoing progress is being made in research and development to ensure the medical industry stays up to date and affords patients with the most beneficial treatments. Answer: Option 2, crops less tolerant to stresses, is the incorrect statement with respect to GM plants. In fact, GM crops tend to be more resilient to environmental stresses, such as storms, droughts, pests and weeds, increasing crop yields. Biotechnological advances have made it possible to produce human insulin in large quantities using bacteria like Acetobacter, Bejetoa, Salmonella, and E. coli. This has drastically reduced the cost of insulin, making it more accessible to those who require it. Biotechnology has had a major impact in the advancement of human health and medical treatments. Eli Lilly, an American biopharmaceutical company, has made significant progress in this field, utilizing human DNA sequences to create and distribute a number of treatments. These include human lipase, insulin, amylase, and urease, which have all been successful in treating various diseases, thus significantly improving the lives of those affected by diseases that can be managed through biotechnology. Biotechnology has had a profound impact on the field of medicine, especially with the emergence of human insulin produced from E. coli bacteria. By combining the distinct peptide, sulfur, hydrogen and nitrogen bonds, human insulin, A and B, chains are manufactured. This is especially beneficial to diabetes patients, as they no longer have to rely on insulin derived from animal sources. In this way, biotechnology is transforming healthcare treatments and providing more effective solutions. Biotechnology is a rapidly evolving field with the aim of transforming the healthcare, agriculture, food and energy industries. It provides a potential pathway to produce insulin for medical use, such as diabetes treatment, through a novel, ancient, conventional or combined method, allowing for stability of both its availability and price. HIV detection often utilizes techniques such as ELISA, PCR, chromatography, and electrophoresis. When performed correctly, these methods are highly reliable and can generate accurate results. To maximize accuracy, it is paramount to recognize the exact method being implemented. Biotechnology is an ever-evolving field that is paving the way for groundbreaking medical breakthroughs. This slide introduces how diagnostic tests can be utilized to detect a wide variety of diseases. These tests are built upon the principle of antigen-antibody interaction, with the most common ones being ELISA, PCR, HPLC and Western blot. Altogether, this slide showcases the capability of biotechnology and its applications and how they are capable of enhancing the lives of people around the globe. Biotechnology has proven to be a substantial asset in the healthcare sector, providing new methods for disease detection. With the utilization of state-of-the-art technologies such as ELISA, PCR, electrophoresis, and Western blotting, the emergence of pathogens can be detected before the emergence of visible signs. It is imperative to detect the disease in its infancy, as early diagnosis often leads to simpler and more effective treatment plans which may reduce the chances of further complications. Biotechnology has had a profound effect on how medical practitioners identify and treat illnesses. Polymerase chain reaction, or PCR, is one of the many tools employed by biotechnology to identify mutations in genes, which can potentially indicate the presence of cancer. The technique involves the duplication of specific DNA strands, thereby making them easier to analyze. This, in turn, facilitates quicker diagnosis and can lead to more effective treatments. Biotechnology and modern science have enabled us to diagnose genetic disorders rapidly and accurately. ELISA, PCR, DNA fingerprinting and Western blot are efficient diagnostic approaches which can be employed to identify and diagnose genetic disorders. These techniques have made it possible for medical professionals to identify and treat genetic disorders more effectively. Biotechnology has made remarkable advances in criminal cases, including 4 major techniques. ELISA, enzyme-linked immunosorbent assay, PCR, polymerase chain reaction, DNA fingerprinting, and HPLC, high-performance liquid chromatography. ELISA detects the presence of an antigen or antibody in a sample. PCR amplifies single or a few copies of DNA for further study. DNA fingerprinting compares an individual's unique DNA signature to a database of potential matches, and HPLC separates, identifies, and quantifies components in a mixture. When used together, these 4 techniques are invaluable in criminal investigations. Biotechnology has experienced an increase in its applications, with the use of transgenic plants being one of the most noteworthy. In order to create these plants, a vector is necessary. The most common vector employed is the T plasmid of Agrobacterium tumefaciens, though plasmids from species such as E. coli, Bacillus thuringiensis and Salmonella can likewise be used. Biotechnological advances over the past few years have substantially improved crop production. Transgenic tomato plants have been engineered to resist various bacterial pathogens, such as Pseudomonas, Phytophthora, Xanthomonas, and Agrobacterium. Thanks to this enhanced resistance, crops can be grown in areas threatened by the spread of these pathogens, thus enabling efficient food production in a more varied range of climates. Biotechnology has enabled farmers to cultivate crops in much less time and with far fewer resources thanks to the development of the flavor-saver tomato. Highly resistant to bruising, bacterial infection, insect infestation, and fungus, this tomato has revolutionized the agricultural industry, showing the power of biotechnology. Biotechnology has enabled the development of genetically modified organisms, GMOs. One of its most significant accomplishments is the production of transgenic golden rice obtained from type A variety. This rice is specially enriched with vitamin A, making it a potent supplement when consumed with a balanced diet. Biotechnology has enabled the engineering of plants with advantageous traits. An illustration of this is the creation of transgenic plants which aid in preventing night blindness. These crops include flavor-saver tomato, transgenic potato, golden rice, and transgenic cotton, all of which have been developed to deliver a positive effect on those consuming them. Biotechnology has enabled the engineering of crops that demonstrate tolerance to certain pests and diseases. An example of this is Roundup Ready soybean, evidencing resistance to glyphosate herbicide. This has allowed farmers to save time, money and improve the yield of their crops. In addition, biotech has enabled the development of insecticides, bacteria and fungi to further protect their crop. In this way, biotechnology has become a beneficial tool not only for farmers, but for society as a whole. Biotechnology has unlocked a vast array of possibilities for the production of goods and solutions to an extensive pool of issues, especially in the agricultural industry. Molecular farming is a widespread practice, taking plants and taking advantage of them by turning them into biofactories or bioreactors. This process entails manufacturing useful elements such as food additives, drugs, and enzymes straight into the plant. Then, these are taken from the plant and used as needed. This process of molecular farming is known as cellular farming, and it offers one of its main benefits as being much more efficient and cost-effective than traditional manufacturing procedures. Biotechnology has been used for decades to create new medicines, crops, and tools for use in many industries. These technologies can revolutionize the biochemical industry and farming by increasing crop production and introducing cellular and molecular farming. Cellular farming uses the energy of cell cultures to produce commercial products, while molecular farming uses genetically modified organisms to produce therapeutic proteins. Bioremediation is also a potential revolutionizing technology, as it uses microbes to clean up contaminated areas. All of this offers the potential to revolutionize the biochemical industry and farming in the near future. Biotechnology has offered new opportunities for exploration in the medical, industrial and agricultural sectors. A collaborative debate is underway on the usage of genetically modified crops as they can have long-term ramifications on the genetic composition of foodstuffs. Keeping this in mind, it is critical to assess the safety concerns posed by biotechnology. Of the 4 assertions concerning the safety issues of GM crops, the one that is not a fear is Statement 1 as gene pollution is a reality that must be addressed. Biotechnology has had a major impact on the agricultural industry through the integration of genetically modified, GM, crops. GM crops tend to possess superior features when compared to non-GM varieties, notably higher yields and improved nutritional value. Despite the potential benefits that GM crops can bring to agriculture, there are several issues that need to be addressed surrounding its use. Chief amongst the concerns is the biosafety aspect. Any alterations to the fundamental structure of the crop can lead to a range of possible consequences, such as side effects on humans and animals, untoward changes to the natural evolutionary process, and so on. It is therefore essential to consider all risks involved before GM crops are put into production. Biotechnology is employed in agriculture, medicine and the environment. This slide focuses on a specific element of biotechnology research. Who takes the decision on the validity of the research? The 4 answers provided are GEIC, ICRISAT, IRI and GAAC, all of which are government or global entities responsible for regulating biotechnology research. These are the organizations that must evaluate the scientific evidence to come to a reasoned verdict on the validity of biotechnology research. The patent rights on basmati rice were granted to an Indian company through the U.S. Patent and Trademark Office in 1997, illustrating one of the numerous applications of biotechnology. Research and development keeps on creating new prospects in this area. India produces a large amount of basmati rice, a type of rice that is popular for its taste and scent. There are currently 25 known types of basmati rice grown in India that are split into 3 categories: white, brown, and red basmati. Each kind has its own flavor and texture, making them highly desired for a variety of recipes. A variety of basmati rice was released into the market by an American company in 1985. This variety was derived from Indian farmer varieties, not those from other countries such as China, Mexico, or Malaysia. This showcases the usefulness of biotechnology in providing new plant varieties with qualities that make them more useful or more valuable. India is renowned for its vast and diverse biodiversity, especially in terms of grain crops. Rice is an especially important one, with an astonishing 400,000 varieties cultivated only in India. This great variety of rice strains allows it to be grown in different climates and lands, making India a leading producer of rice, second to none in the world. Biopiracy is an issue that can't be ignored, as it has serious consequences on both ecosystems and communities. Taking genetic material from countries without their consent is ethically wrong, and it can cause biodegradation, meaning resources are damaged and aren't available to support life. This is why it's so important to protect and utilize bioresources responsibly, not only for the environment, but also for the people involved. We must be mindful of the effects our actions have on others, and take precaution to ensure that we are not taking advantage of a bioresource. Meloidogyne incognitia is a species of root knot nematodes that are major agricultural pests. This species infests the roots of cotton, tobacco, rice and wheat, resulting in significant yield losses and decreased crop quality. To protect their crops, farmers need to take appropriate control measures against this root knot nematode. Biotechnology is a field that's rapidly expanding, offering a variety of solutions to the issues that industrialized nations face. Solutions like these can promote a healthy economy, while preserving our natural resources and safeguarding biodiversity and traditional knowledge, which are essential for securing a sustainable future. The answer to the dilemma, then, is both 1 and 2. An amendment to the Indian Patents Bill has recently been passed by the Indian Parliament. This amendment provides new rules and regulations for filing patents in India with the aim of making it simpler for inventors and companies to safeguard their inventions and products. The amendment covers a wide range of topics, from the sorts of patents that can be filed to the procedure and timeframe for filing. With this amendment, India now has a better arrangement for encouraging innovation and backing the development of technology. Bt cotton is a transgenic plant designed to be resilient to a wide array of pests, such as fungus, bacteria, nematodes, and insects. By utilizing this type of biotechnology in farming, it can lower the need for pesticides, thus protecting the environment and the well-being of people living near the crops. Biotechnology is an ever-expanding area, with a range of practical applications. This slide focuses on the use of natural plasmids in genetic engineering. Plasmids are known to exist in bacteria such as Phytophthora, Pseudomonas, Agrobacterium tumefaciens and Bacillus thuringiensis. These plasmids have been subject to extensive research and are being utilized to create new treatments and treatments. Biotechnology is a powerful tool that has enabled us to construct new organisms with multiple capabilities, one example being the genetically engineered E. coli bacteria. These engineered bacteria are capable of producing a variety of therapeutic proteins such as growth hormones, human insulin, antibodies, and interferon. This biomanufacturing process has become commonly used in many commercial applications. Golden rice, obtained from Taipei, demonstrates the capability of biotechnology to effect change. It is a genetically modified form of rice, enriched with vitamin A, that can help reduce the risk of vitamin A deficiency in vulnerable populations. This type of rice holds great potential for reducing malnutrition and improving global health. Biotechnology has had a significant effect on various facets of our lives, from medicine to agriculture. The Bt gene, which creates a protein harmful for insect larvae, is a prime example of how biotechnological research has improved our understanding of pest control. This gene has been identified in many crop plants, including corn, cotton, and potatoes, drastically lowering the requirement for chemical pesticides. Moreover, Bt gene-altered crops have the potential of increasing crop yields and protecting biodiversity in an agricultural setting. These possibilities are almost inexhaustible, showcasing the full abilities of biotechnology and its applications. Biotechnology is a rapidly developing field with a wide range of applications in many industries, including food production. An example of this is the golden rice variety, which has been genetically modified to contain higher levels of vitamin A. Additionally, various transgenic tomatoes have been created, the most well-known example being the flavor saver, which has been altered to remain flavorful yet have a longer shelf life. Transgenic papaya varieties are also being developed to be resistant to certain plant viruses. By harnessing biotechnology, new foods can be created with improved nutritional value, providing more access to healthier food options. Biotechnology has revolutionized agricultural production with transgenic plants being engineered to display certain traits for resistance against biotic and abiotic stresses. Examples of this technology include golden rice containing potential for improved nutrition, basmati rice popularly cultivated for its aroma and flavor, Bt cotton providing resistance to insects, and Bt potato protecting against late blight. All of these varieties have increased agricultural production, food security and nutrition. Biotechnology has made it possible to change the genetic structure of various plants, including Brassica napus. One benefit is the perspective to diminish the cost of producing hybrid seeds. This is done by using male and female sterile transgenic plants, which have been altered to be unable to generate viable descendants. By unifying 1 and 3 in the transgenic strategy, there could be a considerable reduction in the costs of hybrid seed production. Biotechnology is an increasingly important area across many industries. Among its uses is DNA fingerprinting, which allows us to identify unique information about a person by examining their fragmentary DNA pattern. We can gain a better understanding of genetic diversity by employing techniques such as electrophoresis, chromatography, southern blotting, or northern blotting. Bioremediation is a process that involves using microbes, plants and their natural processes to reduce or eliminate environmental pollutants, with the aim of preserving our planet and its resources for future generations. As biotechnology has advanced, it has become possible to utilize these organisms to help make the environment cleaner. Biotechnology is a field of science that holds much potential for using plants and other organisms to improve human health. This slide focuses on biotechnology's application to the creation of transgenic plants that are resistant to night blindness, a form of vitamin A deficiency. Golden rice, transgenic tomato plants, flavor saver, and basmati variety are some transgenic plants that have been developed to contain higher levels of vitamin A. These genetically engineered plants can not only help prevent night blindness in developing countries, but they can also help the environment by cutting down on the need for synthetic fertilizers and pesticides. Molecular diagnosis is a powerful tool in forensic science, enabling the identification of potential suspects in matters such as parental disputes and criminal investigations. Commonly used techniques include ELISA for identifying proteins, PCR for amplifying DNA, DNA fingerprinting for comparing samples, and gene therapy for treating genetic ailments. These techniques all aid in further examining the evidence and expediting the resolution of cases. Biotechnology presents a great opportunity for gene therapy, which is the replacement of a faulty gene with a functional one. This field has seen a tremendous amount of progress in the recent years. Therefore, the correct answer to the question is gene therapy. We can be hopeful of gene therapy in terms of treating various diseases. Biotechnology is a science with a wide range of applications, particularly in the area of genetic engineering. Plasmid technology, which involves transferring genetic material between cells, is used in gene editing, gene therapy, and other genetic modifications. It can also be used to create organisms with desirable properties such as increased yield or disease resistance. Biotechnology has had a significant impact on our lives, enabling the transfer of one or more genes from one organism to another through recombinant DNA technology. This is used in a wide range of applications, from manipulating genes to obtain desired traits in plants or animals to creating vaccines and developing drugs or treatments for medical conditions. Additionally, 3 other technologies have been developed in the field of biotechnology: recombinant RNA technology, cellular recombinant technology, and organ recombinant technology. Each of these technologies offers its own unique benefits and potential applications. Biotechnology is a field of science with great potential, having a number of practical uses. An example of this is creating transgenic plants which reduce the need for chemical pesticides. Varieties such as Bt cotton, flavor saver, golden rice, and soybean GM plants can lower expenses while farming and advance crop yields. Simultaneously, they can help prevent environmental degradation due to the excessive use of chemical pesticides. This technology has the capability to create a more solid and ecologically friendly agricultural system. Answer: the incorrect statement with respect to the characters of transgenic plants is number 4. Increases in post-harvest loss. Transgenic plants are resistant to pathogens and pests, suitable for food processing, and tolerant to abiotic stresses, which reduces post-harvest loss. Biotechnology is a science that is developing quickly, incorporating various applications. One such example is the amplification of nucleic acid with techniques such as MCR, PCR, ELISA, and gene therapy. These approaches can take small amounts of target DNA and vastly increase them, with them then being used in multiple domains. Biotechnology is a science field that is developing quickly, with numerous potential applications. This slide shows the use of nematode-specific genes brought into host plants using a vector. Vectors like E. coli, Agrobacterium, protozoan and Bacillus thuringiensis are used during this process. Every vector has its own preferences and shortcomings, so it is essential to research the most suitable vector for the desired application. Biotechnology has had a significant impact on agricultural development, especially in terms of crop improvement and disease control. For instance, the creation of vitamin A-enriched rice, such as golden rice, IR8 rice, Ratna rice, and basmati rice, has enabled a more nutritious staple food to be available to millions of individuals worldwide. Biotechnology has proven to be a valuable asset in our efforts to secure agricultural sustainability. This slide examines the influence biotechnology can have on the green revolution by improving prospects for a higher crop yield. Out of the 4 examples provided, namely, better irrigational facilities, utilization of improved varieties, land reforms and genetically modified crops, only the latter does not fall into the green revolution category. It should also be considered that while there are numerous biotechnological applications that can be employed to sustain and promote agricultural progress, it is essential to exercise them responsibly. Biotechnology is a rapidly evolving and versatile sector. With a variety of applications, it is now being utilized for a wide range of purposes, from food production to medical research. A famous example of biotechnology is the Bt toxin, a protein produced from the cry gene. Other genes such as NIF gene, alpha gene and beta gene also play roles in the production of biochemical compounds. This is only a glimpse of what biotechnology can do. Answer. PBR 322 is option number 1 and is the answer to this slide. It has been used as a vector extensively to create transgenic plants due to its stability, effectiveness, and versatility. It is also able to replicate and express foreign genes in plant systems and can be used to create transgenic animals. It has also been used to study gene regulation, cloning, and gene expression. Biotechnology has had a considerable effect on healthcare, particularly when it comes to diabetes. Historically, treating type 1 diabetes required insulin to be extracted from the pancreas of slaughtered cattle and pigs. Thanks to the progress of biotechnology, insulin can now be created using bacteria and fungi. In addition, insulin can be produced from human pancreas, assisting in the treatment of this condition. Biotechnology has been widely employed in the manufacture of medicines, such as human insulin. E. coli is the most commonly used bacterium for this purpose. Other bacterial species that can be utilized for insulin production include Bacillus thuringiensis, Agrobacterium and Bacillus megatherium.